All right. This says we're live. So, hello everyone out there. It's me. And I'm on today with Dr. Fazale Rana from the Reasons to Believe, and I'm sure we'll talk about that. Welcome. If you have any relevant questions pertaining to uh, this topic, because we have a couple of different topics that we're going to go through, then feel free to put them in the side chat and we'll see if we can answer any of them. Um, so first of all, welcome. Nice to meet you.
Well, yeah, thanks for having me, Jackson. If I can call you that, right? Jackson? Is that
Absolutely.
what you go by? Okay. Yeah. Well, thanks for having me. I'm looking forward to chatting with you.
All right. Yeah, sounds great. I have this bit to read for you. It says Fazal Rana is the Vice President of Research and Apologetics at Reasons to Believe. He is the author of several groundbreaking books, including Who is Adam, Creating Life in the Lab, The Cell's Design, and Dinosaur Blood and the Age of the Earth. He holds a PhD in chemistry with an emphasis in biochemistry from Ohio University. And so, you can add on whatever you want. Tell us more about your background.
Yeah. Well, as the introduction states, I do have a background in biochemistry. I did my PhD at Ohio University. I did a couple of postdocs, University of Virginia, University of Georgia, working primarily in membrane biochemistry and biophysics, and worked for a number of years after I finished my postdocs at the Procter and Gamble company in research and development before joining Reasons to Believe. And my primary interest today is just showing people that science and Christianity can be compatible, that we have a particular model for how science and faith, salvationism. So that's the particular policy. But the focus of my work is really to show that science and faith are again fully compatible.
Yeah, absolutely. Yeah, I do agree that they are compatible. I think it's a lot of like creationists usually say like, oh, evolution is not compatible with Christianity and vice versa. But there are more Christians who accept evolution than there are young Earth creationist Christians. So you know, which is kind of promising, I think. Or you know, and especially as we move forward. And hello, everyone in the live chat, Beej and TD Lane. How's everybody doing? Um, so this conversation, in essence, started when RJ Downard, of course, um, tagged me in a link to a post that you had written on regarding endosymbiosis, and that was a few weeks ago. And so, can you kind of give us a your position on endosymbiosis? Do you think that it was, or that there was intelligent design involved? Would you say?
Yeah, I'm uh, broadly familiar with the ideas that have been proposed to explain the origin of eukaryotic cells, uh, through evolutionary processes. Uh, Again, maybe we could broadly call them these category of explanations: symbiogenesis, sometimes endohypothesis, where there's as I understand it, a range of explanations to be compelling. And when I look at the structure and the functioning of the eukaryotic cell, it is the intelligent agent. So I think the origin of eukaryotic cells would be a place where I would see God intervening in the history of life. To me, I think at minimum, the places I would see God intervening would be at the origin of life itself. I think at the origin of eukaryotic cells, the origin of body plans, and I'm thinking more animal centric at this point. And then I would also say at the origin regard to what I would argue is our exceptional nature, our capacity for advanced cognitive abilities. So I would minimally see those being places where it requires a creator's input or intervention.
Okay. Um, hmm. This is. Oh, can you hear me? Can you hear me? My internet is not doing so
Yes,
great.
I can. Can you hear me?
Yeah, I can hear you now. Yeah, my internet's not doing so great, which is kind of yikes. Um, because there was a lot of like a lot of blips uh while you
Mm.
were talking. Um, so yeah. Well, so would you? So with regard to um the to endosymbiotic events, would you say that it was just really the acquisition of mitochondria that uh, that first kind of endosymbiotic event that required intelligent design, or would you also apply that thinking to the origin of chloroplasts and the endosymbiosis of protists by other protists and um, things like dinoflagellates within coral, things like that? Or would you say those only require natural explanations?
Yeah, I mean, to me, you know, if I am tracking with your question, uh, to me, I would say that it would be the origin of uh, essentially the very first eukaryotic cell, as well as the origin of plant cells or cells with the capacity to complex cells with the capacity for photosynthesis. I would see those as being creation events. I mean, can endosymbiotic relationships take place or arise through you know quote unquote evolutionary mechanisms? I think I would be sympathetic to that idea that those could be would be possible. But it was interesting to me, a study done recently by a team that was symbiotic relationship between E. coli and yeast, and they genetically engineered the yeast to be deficient in ATP synthesis, and then they genetically engineered the E. coli to be deficient in thymine production, and so that with the idea that these two cell types would be essentially interdependent upon each other. And but in order for E. coli to be able to benefit the yeast cell, they had to install an ATP ADP transporter that actually would pump ATP from the interior of the E. coli cell to the exterior environment. And in order to do this, they had to essentially uh, make use of that type of an enzyme or that type of a protein from an endosymbiont that naturally exists in nature. And there's other work that I've seen done in the origin of eukaryotic cells that talks about one of the great mysteries is how do you evolve an ATP ADP transporter that pumps ATP from the interior of the cell to the exterior environment. And because this is essentially counterintuitive, there's no benefit that's derived from that direction of transport for a cell. In fact, you'd expect the opposite direction of transport. And so this remains one of the serious challenges for how you would wind up getting, you know, getting an endosymbiotic relationship established. And then, in addition to that, they also had to add genes that encode snare-like genes. And so the point is to get this endosymbiotic relationship established, it required, a fairly sophisticated strategy that relied on foresight in order to. I'm even beginning to wonder, based on that study, if the endosymbiotic relationships that we see in nature actually are pointing to design itself. Again, based on this work that was done in synthetic biology. So you just, the idea of one cell taking up another cell, and there's a permanent residency established. Um, you know, I think uh, raises a lot of questions about how does that mutualistic interaction take place. In fact, the research looking at the, or struggling to explain the origin of the ATP ADP transporter argues that the very first endosymbiont that quote unquote evolved into a mitochondria was actually, or mitochondrion was actually probably an energy parasite. So this idea of this mutualistic beneficial symbiosis is doesn't seem to be a story that actually fits what the data says.
Well, it does seem that yeah, mitochondria is a relative of the of rickettsias, which are parasite, which are intracellular parasites. Um, but they, but I think in a lot of these cases there are pretty clear things that both kind of give off, and it's like the host gives off some metabolic byproduct, and the endosymbiont gives off another, and they both are giving things to each other, and so that's kind of how these things are getting established. Like with the chloroplast, they're giving off metabolic energy, and the endosymbiont is providing them this place to live, and it's also donating molecules that are used in photosynthesis. So I mean,
Yeah, and you know, I'm sorry.
no, go ahead. You're
I,
good.
yeah, to me, I think on the surface, the case for endosymbiosis is not only reasonable. I think there's it's compelling to some degree, because what you're highlighting there are again what appears to be these similarities, and it's not just simply morphological similarities, but there seems to be biochemical similarities that connect mitochondria to alpha proteobacteria, and you also can construct this this narrative as to what would drive this association between engulfed cell and the host cell, and even envision a scenario for how that engulfed cell, the endosymbiont, essentially evolves towards an organelle type status, right? So I wouldn't dispute in any way, shape, or form the fact that there is evidence that favors the scenario that you're describing. But to me, my question becomes, when I start penetrating and looking at the details, and looking at some of the more recent work trying to account for endosymbiosis. So, for example, there's a paper that was published in May of 2018 in Nature, entitled "Deep Mitochondrial Origin Outside the Sampled Alpha Proteobacteria." And part of the argument in that paper is that actually it doesn't look like mitochondria and have any kind of relationship to alpha proteobacteria. This is, again, kind of the standard story, but they argue that, again, that, that the data doesn't support that evolutionary relationship. And the result of this paper is that the identity of the endosymbiont that quote unquote evolved into mitochondria remains unidentified. And this is rather significant, because you can pick up countless scientific articles, countless descriptions in textbooks, in other, other venues, other scientific outlets that all recount the origin of mitochondria, again evolving from alpha proteobacteria, and yet here's a recent study that says that this standard story doesn't seem to be correct. As an example, and really the evidence that you see in favor of endosymbiosis is essentially shared biological designs or homologies that people argue must reflect an evolutionary history. And part of my
And
argument would be that those shared features could essentially reflect a design. I mean, could be accommodated in a design framework
well,
where,
the shared features of the of which part, the endosymbiont or the host, or
I mean, I would say that everything that you would identify as shared features that, again, you would interpret as evidence for this evolutionary story, in other words, or evidence for common descent, could be understood as evidence for shared designs or common designs, or some kind of archetypical design that was employed in the design of, eukaryotic cells and bacteria.
so.
And go ahead, please.
Okay, so for instance, if you look at the chloroplast, which was pretty quickly identified as like a close relative of prochloron and other cyanobacteria, are you saying that that's just uh, common design that's not reflected? It's all these features that it has, including uh, in its chloroplast genome, are not reflective of descent from a free living cyanobacteria?
Yeah, that would be the argument that I would make. And to me, again, just to appreciate kind of the line of reasoning, it's only been really recently that I've have delved into the whole question of endosymbiosis. So I'm still very much myself, in a learning mode about the different ideas that are out there, and really looking in detail at the evidence that people are presenting in favor of the model. So, I'm still, again, kind of in an exploratory phase. But to me, when I look at what is required in terms of protein, the biogenesis of mitochondria,
Um.
and particularly
Okay, sorry.
the import of proteins and targeting of proteins to different locations in mitochondria. To me, as a biochemist, when I look at that, and then I look at the evolutionary scenario for how that system would have evolved, to me, I think there's just gaping holes in that evolutionary explanation. Uh, Franklin Harold has written a book, from drawing a blank on the name of the book, but it's a book published maybe 2016. It's essentially kind of the story of the origin of life and the origin of cells, eukaryotic cells. But he discusses the whole problem of how do you account from an evolutionary perspective for the origin of the protein biosynthesis that's again the targeting pathway to get proteins into mitochondria. And it's a really well done book. And his point at that, or his explanation there, is that this is really a hard problem, but somehow it evolved. And that was essentially, and again, I respect, this is not to lampoon who I think to be a very fine scientist. uh, you know but this to me sets up red flags. It's like, okay, my intuition as a biochemist is that this would be very difficult to account for from an evolutionary perspective. When I seek out what are the explanations, I see, again, a gigantic question mark. Now, you could argue this is an impenetrable mystery that may always be left unsolved. Does that necessarily mean that the evolutionary explanation is invalid? That not necessarily, but to me, it also says that there's a gaping hole. And then when I look to see, okay, well, what is really the case for endosymbiosis? It's primarily shared features. And you could make make an argument that those shared features, reflect again common design. But then as I explore other things about endosymbiosis, I see that there's other gigantic question marks when we start getting into the specifics of how do you account for the biochemical systems that are characteristic of mitochondria, the direction of the ATP ADP transporter in mitochondria, and the idea that there's not a very clear evolutionary history that can be constructed for how that ATP ADP transporter would have emerged. It's distinct. From an evolutionary perspective, it would have to have distinct history from what those ATP ADP transporters that we see in endosymbionts that currently are found in biological systems. And yet it doesn't have a clear evolutionary history. It's counterintuitive that a system like that would emerge. You know, an example, the TIM protein transport into mitochondria, again, are have shared similarities to the TIM and TOM complexes in chloroplasts, and yet people believe that the evolutionary origin of chloroplasts and mitochondria would be independent. And so we were saying that these identical types of systems would have emerged independently. To me, and with such a high degree of similarity, doesn't seem to fit comfortably. Or the fact that the plasma membrane of eukaryotes is fundamentally distinct from the plasma membrane of archaea. The types of phospholipids are fundamentally different, and the metabolic pathways to make those phospholipids are fundamentally different. And yet what we see is a plasma membrane that looks bacterial, not archaeal, in eukaryotic cells. So it's things like that that, to me, raise very genuine question marks about, is this really the best explanation? And so, to me, then, if the primary evidence is shared designs, why couldn't you understand those shared designs as reflecting a design explanation as opposed to an evolutionary explanation?
Okay. One of the things I do want to point out is that the paper you mentioned on the phylogenetic position of the mitochondrial ancestors, Rational Mind says the paper identifies the mitochondria evolving from a species only just outside of extant alpha proteobacteria. Also, other papers since that one by Martigan et al. In 2018 have argued that mitochondria really did evolve from within alpha proteobacteria, so I'm not sure it's settled yet.
Yeah.
That's
Well, but even
right.
and fair point. Yeah, fair point, and I appreciate that. And to me, the fact that it's not even settled, I think, is disturbing, because what we see presented is a very, what seems to be this open and shut case that it was an alpha proteobacteria that gave rise to mitochondria. And you know, uh, maybe it'll turn out that way, um, you know, but right now it is an unsettled question, and that, to me, again, is problematic, I guess I would say.
Well, I mean, when talking about endosymbiosis, it's not hard to. Oh, whoops. Okay. All right. Well, I guess we're fine now. They say. Well, when talking about the the endosymbiotic process that gave rise to eukaryotes, this occurred, by their general estimations, like 2 billion years ago. So I'm not entirely shocked that we're only looking at the fragments of what happened then. And I mean, there are lots of places in phylogenetics where you have major questions. I've been reading a lot recently about the questions over the earliest animals. Or is the most basally derived animal or extant animal, sponges or ctenophores? Well, that's kind of a question, I think, in large part because there's an undersampling of ctenophores, and you've got lots of, you have a lot of sponges, but not a lot of ctenophores, and there's a lot we don't know about their physiology, and same with, placozoans, and so, yes, Fazale Rana does work with reasons to believe. Have you read the book The Vital Question by Nick Lane?
Yes. Yep. Yeah. It's great book.
Yeah. Is was it the Was it the cell membrane that was like the the opposite version of the phospholipid? Was it that one? It's like you mentioned.
Yeah, yeah, yeah.
Yeah.
So for example, the phospholipids that you see in archaeal are distinct from the phospholipids that you see in bacteria.
Right. Yeah.
And so if the host, so the point is if the host is supposed to be an archaean, and the argument for that is because of the similarity in DNA, in the DNA replication, DNA transcription mechanisms between eukaryotic cells and archaeal. People are arguing that that the host cell was an archaean. Then you would expect that you would see again a plasma membrane that would be archaeal like, not bacterial like. So there's a very significant transition that had to happen during the process of eukaryogenesis, where you have that transition in the membrane
Mm-hmm.
composition. And it's not just me, but there's people that are working on this problem that are committed to an evolutionary accounting of the origin of eukaryotic cells, that readily acknowledge that this is a major issue. Um,
Are you? Would you necessarily say it's an issue or a question, though? I think there are lots of questions. Not sure about issues necessarily. Um, but I don't know. I mean, if I look at like a phylogenetic sequence that's very much a ping pong match at the moment, I wouldn't necessarily say that's a problem for evolution. I'd say it's a question, but not really a problem.
well, I mean, to me, I think what you know, if it was just the question that we don't really know the identity of the archaeal host, or we don't really know the identity of the bacteria that produced mitochondria, I would hardly point that out as being something that raises fundamental questions to me about, again, eukaryogenesis. But
Sure.
when I see, for example, gaping holes in trying to envision the pathway by which something like, again, the protein targeting and the protein sorting that you take, that is required for protein biosynthesis in mitochondria, or you try to account for of the plasma membrane in eukaryotic cells. And again, you have experts that are working in this area. To me, that suggests that maybe this isn't the best explanation. Or how do you account for the? So it's the biochemical details that to me are problematic. It's not the overall scenario, or the fact that we don't know certain details about the overall scenario. These are fundamental problems that relate to the very nature of the mitochondria, and some of its most important features, or relate to the fundamental nature of eukaryotic cells that we don't have explanation for. Another piece of evidence that people would point to in favor of kind of the endosymbiont hypothesis is the presence of cardiolipin in mitochondrial membranes. This is a highly unusual phospholipid that's found basically in bacteria, and the bio, and it's also found in mitochondria. So people point to that and say, well, this is clear evidence for an evolutionary history. And yet, again, when you kind of drill down into the details, you discover that the way cardiolipin is synthesized in bacterial membranes is very different than how it's synthesized in mitochondrial membranes. And so
Mm-hmm.
here's a detail that doesn't fit the scenario. That to me those are, again, and so to me the project then becomes, okay, this evolutionary story isn't isn't maybe as well established as I would have initially thought, just based on my educational experience, and just the casual reading that I've done on endosymbiosis, and the arguments for it, and that type of thing. And so the question then becomes, is it possible to explain it from a design framework. And I think it also means that we have to come up with a rationale for why mitochondria look the way that they look. And you were speaking about Nick Lane in his book Vital Question, and I think in that book Nick Lane provides a really elegant rationale for why mitochondria exist in eukaryotic cells, and why um, that if you would design a eukaryotic cell from scratch, that you'd have to have organelles like mitochondria in order to support the biochemical complexity needed for a eukaryotic cell in order for, again, advanced life
Well,
to be possible.
I mean, that seems like a very long end game then, if that's kind of the case. Because, I mean, we're outnumbered by bacteria and they, don't have these systems. Um, Eukaryotes are just kind of the froth on top, But I do also want to point out that Rational Mind also says, for example, there are bacteria type which eukaryotes have phospholipid biosynthesis genes present in Asgard Archaeota. So a group of archaeans that are closely related to or that are thought to be closely related to eukaryotes.
Yeah, I've actually read the paper making that argument, and it's at best a circumstantial argument. If I'm thinking of the same paper that is being referred to here, there's a claim that what is it, the Loki,
Archaeota, yeah.
what is the yeah, Loki Archaeota, yeah, thank you, is is the and again, it's actually not a an individual organism. It's a broad category of
Well,
organisms,
yeah,
and,
it's
and so
a phylum. It's a whole like super phylum.
it's a it's
Yeah.
it's a yeah. So to me, yeah. So something from that phylum presumably was the host cell, but I'm not completely convinced that people have established that you do have indeed the this the biosynthetic machinery to make bacterial type lipids in that. I've read a paper and I found the argument to be very to be circumstantial and very weak
And
at best.
this, they say, Nestlig says another paper bridging the divide: bacteria synthesizing archaeal membrane lipids.
Yeah, I've read that paper. In fact, wrote a blog response to it. Now, apparently, because in this, up until recently, I guess recently somebody's been able to culture um, a Loki archaeota in a laboratory environment, but up until that point, the it was essentially the technique that Craig Venter developed. Sorry, as I'm getting older,
Oh, you're fine.
I'm forgetting terms or struggle with terms
You're
sometimes.
fine.
But Craig Venter, where you basically you sample DNA from the environment, right, and then you reconstruct genomes of these
All
organisms
right, yeah.
from
The
the
eDNA or
using
whatever.
shotgun sequencing and the
Yeah.
shotgun assembly. Yeah. So I mean, Loki Archaeota was essentially discovered that way, and it was so it was kind of a genome that was constructed from from metagenomics, from a metagenomics tech approach. A lot of difference as to what its biochemical capabilities would be from analyzing the genome, where you have a significant portion of the genome that is actually not understood, right? And so it's hard to argue from that analysis, whether or not, again, you actually have that machinery in place
Oh, are you still there?
to actually produce. It was a circumstantial argument that was made, and it wasn't the full. Yes, it wasn't the full
Okay.
ensemble of machinery. It was essentially pointing to a couple of enzymes that they argued could potentially have bridged the gap. But I actually wrote a blog piece where I engaged that particular paper as part of a broader argument.
I do. I mean, I do enjoy discussing endosymbiosis, but we're nearly half time and we haven't talked about anything else. So I do want to. It is an interesting topic,
Yeah.
and I do want to ask. Well, really, the questions that I had intended on on asking in the first place.
Right.
Um, so, sort of a broad question is: do you think that there is a uh, lot, or there has been a lot of design intervention in the evolutionary history of life, or do you think that it's not been a whole lot? What are your thoughts on that?
Yeah, that's a great question, and I mean, just to help kind of set the context for to, for that question, and I don't want to go into a monologue here, but I was an agnostic when I started graduate school. I fully embraced an evolutionary perspective on the origin, the design, and the history of life, and it was really studying biochemistry that convinced me there must be a mind behind at least life's origin in fundamental design. And for a number of years, I would have argued that it was at the origin of life where you saw a divine intervention, if you will, and that the history of life was essentially an evolutionary history of life where a creator either deployed evolution as the means to create or just let the creation unfurl on its own. Uh, and it was really in um, later, it was a number of years later that I began to genuinely question at least certain aspects of the evolutionary paradigm itself. And so, to me, I think when it comes to eukaryogenesis or the origin of body plans, again, the origin of humanity, these are places where I don't think we've got compelling evolutionary explanations, and these are also places where Eugene Koonin refers to them as biology's Big Bang. So at minimum, I would see God's involvement in these places, but you know, but yeah.
I'm
So that would
familiar
be at
with the
least the
paper
minimum place.
where he does say that, and I did actually, and I did do a reference to it in a recent video. Um, I have actually did I did I have actually
Hmm.
done videos on because he says like the Cambrian phyla plant or flowering plants, mammals, humans, and I've done videos on a bunch of those. But of course, we won't have time to get into all those. Um, but yeah, those are yeah the big biological big bangs. Um, do you would you say then that you would say that like you accept speciation, or would you say that
Oh yeah, I
that's
mean,
kind of a base?
yeah, we were talking, before the show. I'm an old Earth creationist,
Mm-hmm.
so I readily accept the antiquity of the Earth and the antiquity of life on Earth.
Mm-hmm.
Um, I see the fossil record as a real proxy for the history of life on
Okay.
Earth. Um,
Okay. I gotcha.
I see that as fully compatible with a historical reading of Genesis 1, where
Right.
day is a period of time. I think the sequence of events that you see in Genesis 1, at least in a broad sense, correlate with the Earth's history and life's history. It's a very scant description of life's history and Earth's history in Genesis 1, but I think there's a really provocative correlation that you see there.
Okay.
Uh, but as an old Earth creationist, I do believe that God was involved in a direct personal way, uh, in creating the history of life. But I also see it not just either or. I see it both and. I think there's both divine intervention and there's also process.
Right.
And so I have no issue with the idea that, I have a peculiar way of categorizing the scale of evolutionary processes. People get troubled by that, but I don't. Um, but it just helpful. It's a helpful device for me to communicate how I think about it. So
Mm.
just take it for that. So for example, I see ample evidence that there is variation that happens within a species. I would dub that microevolution. I know that that concept, distinguishing micro and macro, leaves a bitter taste in people's mouth. For the sake of convenience, I would call it that. Amply, readily accept speciation. I think evolutionary processes would even happen at the genus and family levels. I would simply point out that some of those biological categories are are categories of convenience. They don't necessarily or aren't necessarily communicating anything fundamental. I think speciation is. I think phyla communicates something fundamental as a category, but I think these other categories are arguably of convenience. I would argue I'm skeptical that evolutionary mechanisms can generate genuine innovation. And where that innovation would take happen, to me, it might be at the level of an order. If you think about mammals, or at the level of a class,
Oh,
so it's somewhere in there that I would see God intervening. So I kind of see God as creating basic designs, being responsible for life going from one regime of complexity to the other.
mm-hmm.
Um, and then once that happens, those designs would then vary naturally through evolutionary processes. But where the line is, I don't know that I've got a hard, fast line.
Okay. So if I were to say, for instance, because Kunin says that the origin of mammals is one of the Big Bangs, would you say, or you say that you think there is a purely naturalistic explanation for the origin of mammals, like from the evolution from therapsids and earlier synapsids, the mammal-like reptile group?
Yeah, well, I mean, to me, when I see things like radiation events, I see those as places where it looks like to me a creator was involved.
Well,
Um, you
interesting.
know, yeah. So I've not probed the you know I've not you know the history of life on Earth is a vast is a vast topic, and so
Yeah.
I've not probed in specific details the question of mammalian origins, the origin of humanity. Uh, you know, and I'm just beginning, as I mentioned, to get interested in the origin of eukaryotic cells. So that's where I would feel most comfortable in terms of making hard and fast statements. But when I see radiation events, that to me is suggestive of a creator's involvement. Um, but I would hesitate to go much further than that. If that you know,
Well, I mean
yeah,
the origin of birds is kind of at a radiation of Maniraptor theropods, as well as a number of other organisms like arthropods, uh,
right,
at the end Jurassic, which seems to have been based on molecular studies to have correlated with the origin of angiosperms.
right, right.
Does that, do you see design in that?
Well, uh, kind of to take a step back, and so you can kind of understand a little bit about how I'm thinking.
Okay.
Um, if you allow me, first of all, I would bring up this idea from the philosophy of science called underdetermination theory. And the idea behind that is that theories are underdetermined by data, meaning that you could, in reality, have radically different theories that could account for the same set of data. And I also recognize that science has been, or at least the philosophical framework that undergirds contemporary science, is methodological naturalism, which, as part of that idea, you exclude um, a priori any kind of design explanation, any kind of teleological explanation. Uh, and there are very good reasons why methodological naturalism is a productive way to do science. But I think personally, it also sets in place sometimes restrictions that prevent exploring possible explanations that could sure. And I will point out that prior to Darwin, biology was teleological in its construct. And so it is possible to have biology that's teleological that could be a productive enterprise. So with those two kind of background statements, to me, my project is not so much trying to argue that the data that we see on the table is incompatible with an evolutionary history of life, but rather my project is, if we if we believe that a creator has played a role in a direct way in the history of life, can that approach to biology accommodate the data that we see? In not in an ad hoc way, but in a way that's coherent, that logically is a logical entailment of those ideas. And so to me, I would look at, again, the history of life through the lens of design, and would argue that in a broad sense, the shared designs that we see could either reflect an evolutionary, history, or it could reflect, again, an archetypical, the deployment of archetypical designs. When we see the fossil record, we do see instances of mosaic forms very clearly. And could we understand those mosaic forms as transitional? Sure. Or could we also see them as being, again, reflective of the work of a mind, recognizing that as human designers, we oftentimes will create things that are mosaics that rely on a combination of features that make that particular device or object ideally suited for the environment, or for its, for the demands on that device or that object. And so could you see, mosaic forms that you see, like the therapsids, as you're pointing out, as being mosaic designs as opposed to transitional forms? So to me, my argument would be, I think you could just as readily explain the history of life on Earth and the design of life, or the diversity of life, from a design framework where imposed upon kind of a creator's involvement are natural process diversifications of those core designs. So that's kind of the way I would approach it. But ultimately, what motivates that to me is what I see it to be. I think the fact that the origin of life to me looks like it is clearly open and shut, a case of divine intervention. And the design of biochemical systems to me seems to be open and shut, real evidence for design, or for the work of the mind, and that then shapes how I see the history of life. So I don't know if that's helpful to you, uh,
I
or
yeah,
or
I
more confusing.
I mean, I see what you're saying. But I have to ask then, why is it that the evolutionary model is predictive of these transitional forms, whereas the created mosaic idea really isn't, or hasn't been?
um, uh, the created mosaics are not predictive of. I'm sorry. I'm not quite sure I know what
Oh well,
you're asking
if
there.
well, evolution or models within evolution make specific predictions about where certain fossils should be in the like the lineage of birds, for instance. Researchers predicted that there was a four-winged stage in bird evolution, which we now have Microraptor, which is which demonstrates that it had the four asymmetrical wings. Why is it that evolution makes these sort of predictions, or Tiktaalik another example, or within the therapsids is like the double hinge jaw joint? Why does evolution make these predictions? Where are the same predictions that this created mosaic idea would predict? Where are those sorts of forms? Or is that did I
Yeah.
help at
Okay.
all?
I see. Yeah. No. 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 I see. No. No. That helps a lot. Yeah. Well, um, I mean, it is true, for example, that the evolutionary model would make predictions as to where fossil specimens ought to be discovered. But there's also instances where I think those predictions are also represent failed predictions. So, for example, I was just reading um, work done, published PNAS about for bats and moths, where the evolutionary argument would be that moths evolved hearing organs in order to be able to detect the ultrasonic sounds of bats, right, that were preying on these moths. So these hearing organs allows them to escape the echolocation capabilities of microbats, right? and and so you would predict from an evolutionary perspective that when you go into the fossil record looking at lepidopterans, that you would see essentially the origin of hearing organs after the point where echolocation appears in bats. And I think off the top of my head, the bat specimen, that fossil specimen that shows evidence for echolocation, is close to about 50 million years ago, something like that.
Well, yeah, the
And
oldest ones
so
still show
what we're
echolocation. Yeah.
seeing is that the evidence for, yeah, but what you end up seeing is that there's evidence that lepidopterans had hearing organs about 90 million years ago. And so there's an example of where you've got an evolutionary model that then essentially makes a prediction as to where you would find certain lepidopteran fossils, and you actually find them in a different location, or the transition from a nocturnal lifestyle to a diurnal lifestyle in lepidopteran, again, presumably was explained as being a coevolution where some of the moths adopted a strategy of becoming diurnal to escape predation from bats. And yet again, we see that transition actually, predates the origin of echolocation. So here's two examples where you'll see evolutionary theory making predictions about where fossils should be discovered that turn out to be,
Well, sure. Well, I mean,
quote unquote falsified. Now you could, right? Okay, so the point is, as I don't think it's, or like you talk about tiktaalik. Is that how you pronounce it? Tiktaalik, tiktaalik.
That's how I do it.
I never know how to
yeah.
pronounce it, but tiktaalik. Okay, so this is supposed to be this perfect transitional form between, again, a lobe-finned fish and the very first tetrapods. Well, you actually have evidence for tetrapod Acanthostega or Ichthyostega footprints at 400 million years ago. So
Yep.
15 million years prior to. So right. So that's an example of where again you see a prediction.
I think that I think you might misunderstand the point of the footprints. Yes, you're right. There are tetrapod footprints that do predate Tiktaalik. Tiktaalik still has all the features that we would expect a fish tetrapod transition to have. It has these intermediate wrists and ankle joints. It has fins, but it also has the evidence of like lungs and stuff, and it has this flattened head. It has all these features. While the timing was off, as we now know from those footprints as well as other finds, it still has the features. Those haven't gone away. They're still very much indicative of the transition. And with regard to your point about some models are within evolution were wrong. They're wrong. We figure out they're wrong. Then you gather more data, and you make new predictions based on the new data. So I mean, yeah, that happens. One of the predictions about natural selection was that it would basically remove all evidence of genetic homologies. Right, there was the universal acid was what they called it, but now we know that's false. Homologies are retained through natural selection. So, I agree.
Well, and just so you understand, right? But just so you understand my point, I'm not saying that when you look at opposing models and then evaluating those models, and some turn out to have supporting data, some turn out to be falsified, and you go back to how science is done. So I'm not faulting or being critical of that happening, or that doesn't, in my mind, count against the evolutionary paradigm. But so often I hear that this claim of yours that that evolution predicts things, and it's very easy to highlight those instances where those predictions, right, but then on the other hand, there are instances where those predictions, where things are where you see temporal paradoxes, where things aren't where they're supposed to be. And so I'm not so sure that the evolutionary paradigm has this really clean, impressive track record of making those kind of predictions. Now, where would I predict mosaic forms to appear in the fossil record? I don't know where I would make. I've not thought about that question, so it's a really good question that you're asking, a fair question. Um, but likewise, if we see mosaic forms in the fossil record, we could easily understand them as being, again, creatures that have been perfectly designed to live at the water's edge, where they would have, again, a mosaic of features that would make them ideally suited or ideally adapted to that environment. So you could understand them, um, you know, or you could accommodate their existence from a design framework.
Um, it I don't know, the idea that they were just created to live at the water's edge seems kind of ad hoc. Um, but uh, I mean, about it's not in, or the things aren't in the right place. Well, that's kind of an issue of taphonomy, really. Uh oh. Can you still hear me?
Yeah, I can. Yeah.
Okay. Well, uh, the there are problems with fossilization, and so they predict approximate locations where these things should exist, like the ancestor or the fish, the fishopods should be in the Devonian, and they are. Eusthenopteron, Panderichthes, Ichthyostega, Acanthostega. These whole series is in the Devonian. Oh, what the footprint showed was that the timing was different. It wasn't in the middle to late Devonian. It was the early to middle Devonian, and so we still have these fossils documenting this transition of characters, but it's like now it's really just the timing that has kind of shifted. Um, Same with mammals. It's like, yeah, the mammals have a huge radiation after the dinosaurs died off, but they were around, back in the Triassic, and the mammal-like reptiles predate the dinosaur or the little pre-dinosaurs. So um,
Well,
go ahead.
I've often wondered. I'm sorry. Yeah, I've often wondered, how much of uh, how much looking at the fossil record from an evolutionary framework is actually retrodiction more so than prediction. I mean, so do we believe that, is there something fundamental in evolutionary theory that would say, that the first tetrapods would appear over 380 million years ago, 360 million years ago? Is there something fundamental in evolutionary theory? Or are we saying that because we see a fossil record that shows this is where the first tetrapods appear, right? Or is there something fundamental, saying that we expect a mammalian radiation after the extinction of dinosaurs? Or is this something that, again, what we observe, and then we go backward and talk about this as being something that the evolutionary framework would predict? If we just simply took the diversity of life that we see today, and try to construct an evolutionary history or sequence based on that, would we actually, again, predict the sequence, or is it a retrodiction of sorts? And that's something, regardless of that's just a question that I have, not with any kind of agenda behind it. Is how much of the claim that the fossil record is exactly the way what we would predict, is that really a genuine prediction or a retrodiction?
Well, I'm not so sure. I mean, if you look at, like, go back to Microraptor, researchers had a very poor fossil record of the bird dinosaur transition for a long time, and Microraptor was, or a form with what they called it Tetrapteryx. A four-winged form was predicted based on how they figured bird evolution should have occurred, like in the early 1900s. But this form, the real Tetrapteryx, wasn't found until like 2001, something like that. And so, I mean, that seems, this is before they had much of a fossil record, and they're predicting a pretty particular set of characters, or at least this one particular character. And then a long time goes by, and then they find it. I mean, that doesn't seem like a retrodiction. Did I cut out?
Yeah. No. I heard it.
Okay.
Yeah.
Same with
Yeah.
like.
No. I
Okay.
see your point.
I would say the same with
Yeah.
the mammal-like reptiles. The double hinge jaw joint was where they had like Dimetrodon, which has the sort of reptilian joint, and then on the other hand you have the mammal the mammal jaw, and they're like, well, as Robert Broom predicted, well, there should have been this double hinged joint as the jaws were going up, or many of our jaw bones were leaving to become our middle ear bones, and so we should expect this particular sort of configuration of bones. And then years later, they found it. So I'd just say that's an example. I was kind of intrigued by what you said regarding you said well, correct me if I'm wrong, that you would suppose design events in you said the major innovations.
Yeah,
Was that
or
correct?
where life goes from one regime. Yes, yeah, life goes from one regime of complexity to another, or there's yeah, innovations, biological innovations.
Okay. So in the if we're looking at tetrapods, for instance, would you say that the origin of the limb is a major innovation, or or would you say it was not?
The
Like to go
origin of the
from
tetrapod
yeah,
forelimb.
to go from like a fish fin to a tetrapod limb.
Hmm. Um, yeah, I would have to. I don't know that I've ever thought about that those terms. That's a good question. Yeah, I don't know. I'm not quite sure. I'd have to think about that a little bit more. Yeah, because in a sense, I think what you're pointing out, which is a fair point, and that is, regime of complexity to the other, or I'm talking about, innovation taking place in the history of life. These are probably fairly ill-defined concepts, and probably they're imprecise concepts. So I would grant your point. I am frankly still exploring where I would where I would not made any kind of hard and fast um, really delineation as to where that. So I would grant your point that maybe the concept of innovation and complexity that I'm deploying are not are not as defined, or are not very well defined, and that creates kind of ambiguity that makes it very difficult maybe to assess some of these claims that I'm making.
Hmm.
And to me, I feel most comfortable about engaging these ideas when it comes to the origin of humanity, or when it comes to
Okay.
um,
Well, we can
the
talk about
origin
that
of
if you want.
life.
If we want to talk about the
Because that's
origin
just
of.
that I mean that's just where that's where I put the bulk of my work and focus on. So
Okay. Well, we can
uh,
talk about the origin of humanity. We only have about 25 minutes left, so we can do that. Where do you see the sort of disconnect between our human lineage and other primates?
I mean, to me, where I see the fundamental disconnect is not so much with respect to our biology, but with respect to our behavioral capacities. And so, to me, I see the origin of humanity, both anatomically and behaviorally modern humans, happening at about 135,000 years ago. And I think both the modern human anatomy as well as the behavioral capacities occur when you see the appearance of a globular skull, in which defines, I think, largely is the defining feature of modern humans, is the globular skull, which is a highly unusual morphology,
Okay.
and the flat the flat face. And this is about the time that you begin to see the flickering of evidence for symbolism in the fossil record, which I would argue is uniquely a modern human capability. And so that's where I would define kind of the the birth of modern humans, and where I think you begin to see discontinuity between modern humans and what precedes modern humans in the fossil record.
Okay. Interesting. So are you kosher with like humans being descended from Homo erectus and Australopithecus, etc.? So that's not really the issue you would
Um,
say necessarily.
yeah, I mean, I would take the view that human beings were created, um, but to me, the emphasis that I would place on that creation event would be, you know, our unique and exceptional nature as human beings, our advanced cognitive capacities that I think are discontinuous with what we see preceding humans in the fossil record. I mean, I would argue, consistent with my position, that the shared features that we see with humans and Australopithecines or with Neanderthals, for example, um, or Homo erectus, would be examples of common design as opposed to common descent.
Hmm.
To me, I think the place there you'd see the sharp demarcation would be the appearance of the modern, our behavioral capacities.
So so you you would say our similarities with Homo erectus are not the result of common descent. That
No, I would say it's reflecting shared design.
but is there really a necessity for us to have the same sort of teeth and uh, entrance for the or the foramen magnum and pelvic structure and wrists and ankles and all that? Is there really a necessity for us to have the same as these other totally unrelated hominins?
Well, I mean, this question you're asking is a really good question that I've given quite a bit of thought to recently. And to me, the idea of homologies that we see in biology actually make the discipline of biology possible. I mean, if we lived in a world where we didn't have universal biochemistry, we didn't have a universality to cell biology, if we didn't have, again, kind of these homologies that we see in in animals, at least in the tetrapods, um, you know, and I mean, this would broadly apply to the animal phyla in general. But if we didn't see these types of homologies, then biology would be a very difficult scientific enterprise,
Sure,
because what we would learn about one organism only applies to that organism. So
sure.
it's rather remarkable to me that biology is even possible as a discipline. And so even if you adopted kind of an evolutionary perspective on life, life's origin, life's history, and diversity, it's almost eerie to me uh, that you would see this type of an anthropic coincidence, that the very mechanism that drives evolution is actually what also makes biology possible as a discipline and the discovery of the life's history and life's design possible. And so to me, when you apply that then to the question of human origins, you could argue that the shared features actually, again, allow us to have a deep understanding of human biology that would only be possible if that kind of archetypical design existed, right? So that's
I'm not following
so
you
there's
here.
a rationale, right? So
Because
in other
oh,
words,
sorry, go ahead.
no, please ask your question.
Okay, because I'm confused because you're saying this. It makes sense that there is this archetypal design, whatever. But at the same time, we're not related to the other organisms who share it most closely with us.
Um,
Or
I
did I
think
misunderstand you?
your part of the challenge that you might be having, understood me or not, but the thought of Sir Richard Owen, he is considered by many people to be the father of comparative analysis, and was actually responsible for elaborating the concept of homology and proposed the theoretical framework to interpret homologies, where he argued homologies reflect essentially an archetype that exists in the mind of a creator, and he produced
Right.
this essentially model for what, right, the vertebrate archetype would look like.
Right.
And so he saw the archetype as reflecting a teleology, where there was a first cause that created these, again, archetypical designs that then were manifested in the created order. So it's almost like a Platonic approach to biology, adopting that, or that idea and using it as a starting point. Again, what you see is that when you think about things from an archetype, is that you expect to see shared designs in biology, and those designs aren't necessarily reflecting an evolutionary relatedness. These organisms would group together into the same categories, but could potentially have very different, could have independent origin events, if you will, right? And so for me, I would see, that you have the Australopiths, you have the Habilines, you have the Erectines, you have, archaic, uh, for lack of a better term, kind of the would form distinct groups, that those groups would cluster into a larger category. But that just because they cluster into those categories, that doesn't reflect evolutionary relatedness. But if you're thinking about things from the design framework and using archetype biology, then it reflects, again, kind of shared designs that you see in these organisms. And so that's how I would see those shared designs, is kind of from that perspective. So
Um,
so in other words, we're looking at we're looking at the same data from very different theoretical frameworks, if that makes any sense,
okay.
right?
This this sounds, in a sense, and I hope to clarify with this next question, but it does kind of sound in a way like larger baraminology. And I want to clarify. Um, if I were to ask, for instance, do you think that arthropods and cephalopods, are they related to each other? Is there a common ancestor between them?
um, I don't know enough about arthropod or cephalopod biology to answer that question definitively.
Hmm.
I just haven't studied. Um,
Well, that's fine. I mean,
I just haven't,
well,
yeah,
sure. I mean,
those
I don't
groups
expect
enough to.
you to have like a in depth knowledge of every organism and whatnot. I just it but these are different. Well, mollusca is the phylum which cephalopods are within. But do you
Right.
think that do you think the phyla like chordates and arthropods share a common ancestor?
No.
No.
Chordates and arthropods.
Yeah. Can you hear me?
Yeah, yeah.
Okay. It keeps dropping. I'm
You
sorry.
said chordates and
Yeah. Chordates and
you said
arthropods.
chordates and arthropods.
Yes. Would you say that they have a common
Yeah,
ancestor?
no, no, because one's a deuterostome and one's a protostome.
Okay. So can I ask this then? Do elephants and starfish have a common ancestor? Actually, let me back it up a little bit. Do elephants and tunicates have a common ancestor? Would you say that? Because they're both chordates.
Uh, yeah, I would see them as being distinct. I mean, from an evolutionary perspective, the argument would be they do, but from a design elements of their embryological development that is common, but that would just be an archetypical design for embryological development.
Okay. So if I were to ask then, um, tunicates and starfish, would you say that they share a common ancestor because they're both deuterostomes? Can you hear
Beg your
me?
pardon?
Uh,
You cut out for a bit.
okay. Would you say that tunicates and starfish share a common ancestor because they both are deuterostomes?
Um, no, I would see them as distinct.
Mm, interesting. So even though they do have very similar developmental systems, like they both undergo, they become a morula and a gastrula, and they develop their anus before their mouth embryonically. Even though those developmental systems are the same, they're still created separately.
Yeah, I would see them as distinct. You know, yeah,
Um,
distinct creations. Because again, with this idea of the archetype, that the shared developmental processes that they have allow us to study one and then have an understanding about the development of uh, basically animals that would cluster as deuterostomes. That by understanding the embryological development of one, we have insight that allows embryology to be possible as a discipline.
so then,
Something that
oh, sorry.
I yeah, please go ahead.
Oh, I was just gonna say. So, by that same token, though, shouldn't arthropods and mollusks be related to each other since they both have the same developmental program? They're both protostomes.
It's well, I mean, again, if you're the same developmental program doesn't necessarily in an evolutionary framework that implies common ancestry or shared ancestry. In a design framework that reflects an archetypical design
Okay,
that um,
I see what you're saying.
so yeah, so I think now I mean, something that I find intriguing, and I'd be interested in your reaction to this is, I've not studied the Cambrian explosion in extensive detail, but I have, you know, dabbled in it a little bit.
Yeah.
And one of the things that I find in um, to the Shenzhen fauna in China, what you see is the co-occurrence of uh, gnathostomes, chordates, hemichordates, tunicates, echinoderms, and then also kind of this enigmatic phyla, Viticola. But these are all more or less showing up in the same in the lower part of the Cambrian, and yet from a evolutionary perspective, you would argue that that the echinoderms gave rise as a side branch to hemichordates and then gave rise to tunicates, which then gave rise to chordates and then the jawless fish. And so the fact that you're not seeing these appear sequentially, but appearing, essentially, or co-occurring or appearing simultaneously at the very base of the Cambrian, to me is really intriguing, and is why I would question whether those shared features reflect an evolutionary history, or as opposed to reflecting design, because we're not seeing again that sequential appearance of those different forms, but their co-appearance. And again, this is at the very base of the Cambrian.
Mm-hmm. Um,
So
well, oh, sorry, go ahead.
Go ahead.
Well, I do want to point out we only have about 10 minutes left, and I don't want to hold you over.
Sure.
But, well, for starters, there are not gnathostomes in the Changjiang. That's Cambrian. Gnathostomes didn't appear until the late Ordovician, early Silurian. You have sort of the split between the chondrichthyes and other teleosts.
I've
I mean, if there were gnathostomes in the Cambrian, that would be
there's
pretty big data. That'd be pretty big development.
yeah well yeah well there's I mean, I've got papers that basically show you have um, uh, yeah, you have I guess craniated fish in
Well, yes.
the
Well, yeah. There are cephalaspidomorphy. No, I think it's pteraspidomorphy. Is a jawless fish. Is a group of jawless fish that appear right at the end of the Cambrian. So you have the first true fish right at the end of the Cambrian, but they're agnathans. Well, Agnath is kind of is a paraphyletic term.
Oh,
Generally.
that's what I meant. Sorry. Yeah. You're right. I misspoke. I misspoke.
Oh, that's
I
fine.
meant, yeah, I meant Agnathan. Yeah. My bad. My
I
bad.
do agree with you that there are the you have the Agnathans and you have the Vitulicolians and Pikaia and all these guys sort of appearing around well, that's kind of a misspeak. It looks like you have the first appearance of their fossils at that time, and what that means is, well, fossils can only denote the minimum time period that an organism lives by definition. And so molecular clocks show is that the typically the paleontologists take a literal approach to the fossil record, especially with regard to the origin of phyla. Like we find these fossils. This is about when they started, but based on what we know about the molecular clocks, it seems more like this is when they're starting to biomineralize. The Cambrians largely characterized by an origin of mineralizing forms. That's why the arthropods are so huge. They just like out of nowhere. You got anomalocarids and morellomorphs and the lobopodians, and so it's really more of an instance of some of those phyla definitely did appear in the Cambrian, but with reference to chordates, the split between echinoderms and chordates appeared in the late Precambrian, so very late Ediacaran, and then you get, like you said, hemichordates. You have like I don't think it's graptolites, but I do believe I remember reading about hemichordates in the Cambrian. So it's really just more of from what I understand, it's more about their biomineralization than their origination. Because there are actually a couple of different radiations that are appearing around the start of the Cambrian and after the Cambrian starts. There are actually like 2 or 3 different radiations going on there. That's my very abbreviated opinion.
I just and to me, I'm I'm not real impressed, I guess I would say, with molecular clock analysis, and it's not simply from my old Earth creationist perspective that I'm making that statement. Even if I would operate within an evolutionary framework, I'm not real impressed with molecular clock analysis. I think the calibration of clock, the molecular clocks, is really difficult to do
Sure,
and do
absolutely,
well. And
absolutely.
um, so I guess I'm not really impressed with the use of molecular clock data to try to make an argument that there were um, events happening in the history of life that were invisible to us because of the vagaries of fossilization
Well,
and
it's
that
kind of
type of stuff.
necessitated. I'd say it's kind of necessitated. I mean, you have sponges and Nidarians in the Precambrian, but then you don't see them again until the Cambrian. You know, like you have the sponges in the I think the Ediacaran. You know, and then you don't see them for a while, and then you see them again in the Cambrian. Well, there were sponges there. We just don't have them. You know, same with Nidarians. They didn't fossilize unless they were corals or they were in Lagerstock.
But
Go ahead.
yeah, but and that and I have no problem accepting that point because there's something you can point to in the fossil record that that makes that explanation, you know, reasonable, right? That maybe the absence that we see, the absence of fossils, just simply reflects again, uh, the fact that there's just again the vagaries of the fossilization process and the burial, the taphonomy, but um, yeah, but it's just in absence of any of having anything in the fossil record to substantiate the molecular clock claims, I just find it hard to, given again the what I think to be the real limitation, want to see that as being somehow a workaround to what I see to be, I think, a real challenge that the co-occurrence of these phyla represent.
If we did find, say, Pikaia like, or if we found something that was sort of intermediate between, you know, um, I don't even know how that would work, but echinoderms and chordates. Let's just say, just hypothetically, um, in the fossil record. As I said, I don't know how that would work because they're, yeah. But anyways, if we were to find such a critter, um, would that affect your position on the Cambrian explosion at all?
Well, I mean, it would be. I'd have to. I mean, I'd have to think about it, and it would depend on specifically what was discovered. But
Sure.
um, I mean, the argument that I'm making right now to be retracted or withdrawn. You know, um, you know, and to me, I also want to make clear that when it comes to the Cambrian, I'm not adopting a position that all the phyla show up in the Cambrian and there's nothing that shows up prior
Sure.
to that or before that, but rather in this window of time. So it's hopefully it's not an overly simplistic presentation of the Cambrian, but but anyway, that I guess it would depend on the specific nature of the discovery. See in the Precambrian that looked like it was transitional and was then I think then your interpretation would be probably a stronger interpretation than mine.
Hmm. Well, we're just at about the hour and a half mark. So I guess I'll let you have the final word on whatever you want to say, and then we'll call it quits.
Yeah, well, I just appreciate you taking the time to invite me to be, on your program, and enjoyed the conversation a lot. Hopefully, it helps you and your viewers understand my position a little bit better. Whether or not they find my position credible is another question, but at least if you hopefully understand my perspective a little bit better. And what I'm trying to do is come to the table with a scientifically robust perspective on the origin and the history of life that is again shaped from a creation slash design framework, um, and hopefully taking the data seriously and not sidestepping data that is quote unquote inconvenient. So I appreciate you giving me a chance to have a hearing. We'll to do this again. It might be
Sure.
fun maybe to do this conversation again, but maybe focus on one particular area of life's history, so that we're uh, you know both well versed, I guess, or better versed than I am on some aspects of life's history.
Sure. Sure. That's fine. Yeah, we can always do that. I enjoyed this too. Um, if you could stick around for just a minute after I close off. As for everybody else, thanks for watching, and we'll see you next time.